We talked about fatty acid degradation, right? So that's how you get energy from lipids, okay? So now we're gonna talk about how do you make those lipids and what are the chemistries involved, okay? All right, so again, I start with this nice picture of a chocolate cake. Just to remind you that the last thing that we talked about in our previous lecture was if you have both fats and sugars, and fats typically has two directions that they can go in your metabolism. Either they can be stored or they can be degraded to acetyl-CoA, okay? However, if at the moment of your fat intake you have a lot of sugar, then what the sugar can do is remember that the sugar becomes acetyl-CoA, and acetyl-CoA becomes this thing called the malonyl-CoA, and then malonyl-CoA inhibits this thing, okay? And you should know what malonyl-CoA inhibits, okay? Like which part of fatty acid degradation that it really inhibits, all right? If you have sugars, so you don't break down lipids, and in the end, you're going to store lipids, okay? So today, my focus is on this part, okay? How do you go from this thing called the malonyl-CoA and goes to fatty acid biosynthesis, okay? So we know that malonyl-CoA is this thing where you have it, then your fatty acid degradation will stop. And it is malonyl-CoA that you use to synthesize your lipids, okay? Okay, so we want to know this guy. All right, so first of all, give you an overview of fatty acid biosynthesis. Now, animals can readily convert carbohydrates to fats, okay? However, we cannot really carry out the net conversion of fats to carbohydrates. Why is that? We don't have glycylation, exactly. Because we don't have glycylation, so we cannot convert acetyl-CoA to pyruvate, okay? Now, fatty acid biosynthesis occurs through intermediates very, very similar to those of the fatty acid oxidation, okay? So if you look at the chemical structures, they're going to be like almost the same, right? Just like when you are doing glycolysis and gluconeogenesis. The intermediates are pretty much the same thing, however, they use different enzymes, right? Now, in the case of fatty acid biosynthesis, there are several things that are different between synthesis and degradation, and this you have to know, okay? Number one difference is your electron carrier. In biosynthesis, we are going to use NADPH, okay? Whereas in degradation, we use NAD and FAD, and we generate NADH and FADH2, okay? But in biosynthesis, we use NADPH. Now, remember I did talk about this at the very, very beginning of our class that NADPH usually is used for biosynthesis to make stuff, NADPH. To degrade stuff and translate that stuff to energy, NAD, okay, and NADH. Okay, so NADPH usually used for biosynthesis, whereas NADH is used to make ATP. Okay, so this is biosynthesis, so we're going to use NADPH. Second thing that's different is that we're going to have a carboxyl group activation, okay? In this case, we're going to use this thing called malonyl-CoA, and malonyl-CoA is used as the carbon addition unit instead of acetyl-CoA, and I'll show you what that means in just a minute. Okay. The building block for fatty acid is malonyl-CoA, okay? It's activated form of acetyl-CoA. The acyl carrier is different, okay? So for beta oxidation, we're using coenzyme A. Okay. However, for fatty acid biosynthesis, we're going to use this thing called the acyl carrier protein. Okay, acyl carrier protein. In short, we just call it ACP sometimes. Okay, so it's not a coenzyme; it's a little protein, and on the little protein, you will see in just a minute, it has a modification. All right, and one of the last thing, the fourth thing that's different is the stereochemistry. Okay, now. previously when we were talking about fatty acid oxidation, I didn't really stress on stereochemistry because there's really no need to do that. However, now when we're talking about fatty acid biosynthesis, this is where I want to emphasize on the fact that they are using different stereoisomers of the 3-hydroxy thiol esters. Okay, fatty acid degradation 啊，我们记不记得它是 alkane hydroxy keto 嘛，对不对？但是在 hydroxy group 的那个时候啊 ，OK， 它就会等于有4个 substituents， 所以你就会有一个chiral center。OK， 那但是在 degradation 的时候呢，那个 hydroxy 都是 S form 的 ，OK，S form 或者是 L form， 随便你要叫 S 或 L 都可以。Right？ 但是在我们要做 fatty acid biosynthesis 的时候，合成的时候，它的 stereochemistry 会是 R form 或 D form， 随便你 ，OK，R 跟 D，S 跟 L。Biosynthesis 的时候是 R form，degradation 的时候是 S form。OK， 这是一个小小的不同。那为什么会有这样子的落差？就跟它 enzyme 有关嘛。OK。All right. The very last thing, this doesn't apply to bacteria. Okay, it only applies to eukaryotes. Is that the cellular locations in which these things happen is different. Okay, fatty acid degradation occurs in the mitochondria. Yes, and fatty acid biosynthesis occurs in the cytosol. Okay, now in a way that also makes sense. Okay, why is that? Okay, why is that? In the mitochondria, what's there? 什么东西很多在 mitochondria 里？可能 TCA cycle 的 intermediates 很多啊 ，TCA cycle 的 enzyme 很多啊，ETC 有没有？那些东西都在 mitochondria 里。好，那如果你说今天 we're gonna let's say hypothetically we make our fatty acid inside the mitochondria, okay？ 那但是问题是说我接下来要把这个 fatty acid 储存在某个地方，或者是拿出去输送。那如果我放在 mitochondria 里面，那它就有2道 membrane， 就是2层要 penetrate， 它要 penetrate 从 mitochondria 里面出来到 cytosol， 然后要从 cytosol 里面出来到外面，它就会有2层要去 penetrate。那其实你在想，这个不太合理嘛？因为如果我们是要储存 fatty acids 变成 TAG， 然后或者是要把 fatty acid 传给别的细胞使用的话，你不太会在 mitochondria 里面去做。OK，mitochondria usually 就是很多这个 TCA cycle 的 enzyme， 很多这个电子传递链的 enzyme， 那通常是会把 acetyl-CoA degrade 掉。OK， but fatty acid biosynthesis, you want to make fatty acids. Okay, you want to make it. Therefore, you're gonna do it in a cytosol. Right? So these are the 5 things that are different between synthesis and degradation. Okay. Cellular location of lipid metabolism. Okay, just give you a pictorial view. We have animal cells and plant cells. Okay. Now in animal cells, your fatty acid degradation, acetyl-CoA production, ketone body synthesis, these things all happen in the mitochondria. Okay. Now if you want to synthesize lipids, okay, so you're going to do it in cytosol. Okay. We're going to make fatty acid biosynthesis. Here in the cytosol, you have a high NADPH to NADP ratio. Okay, so most of the NADP is in the reduced form. Okay, so that's what it means. And in the cytosol, we also have an active pentose phosphate pathway and the malic enzyme. So we're going to generate NADPH outside. We don't generate NADPH as much in the mitochondria. Okay? All right? And there's this little thing called the endoplasmic reticulum, ER. You know, you guys probably know ER more than I do, you know, because I don't know what it is. All I know is this ugly thing that's flying around the nucleus. But yeah, this ER is going to be a little bit interesting to us because we're going to talk about those two, fatty acid elongation and fatty acid desaturation. Okay, we will talk a little bit about that, but not very extensive. Okay. In plants, what you have, okay, is turns out your fatty acids are generated in the chloroplast. Okay, that's a very interesting difference between plant cells and the animal cells. Okay, your fatty acid synthesis occurs in the chloroplast. Why do you think that's the case? Why do you think for plants they will actually want to do fatty acid synthesis in chloroplast? Probably has something to do with NADPH. Okay, because in the chloroplast, that's where you have your oxygenic photosynthesis, right? And the end result for oxygenic photosynthesis is NADPH. Okay, So in the chloroplast, you have a bunch of NADPH. Therefore, probably it's a good idea to use that for fatty acid biosynthesis. Okay? All right. And a little blurb down at the very bottom. I don't know if you can see that. It says bacterial cells have no localization. I mean, okay, that's like, duh. Okay? All right, now, however, acetyl-CoA, okay, we know fatty acid is synthesized from malonyl-CoA, but malonyl-CoA comes from acetyl-CoA, okay? Now, here's the problem, okay? What is the problem? The problem is that for most non-photosynthetic eukaryotes, okay, like us, we're not photosynthetic, nearly all acetyl-CoA is generated in the mitochondria. By which enzyme? Yuan. Pyruvate, 叉叉 carboxylase 不是吧 ？Pyruvate 什么？哈 ？Pyruvate synthetase 哦，我要做 acetyl-CoA 啊，我做 Pyruvate synthetase 也没有这个东西 ，Pyruvate synthetase。Pyruvate 到 acetyl-CoA 会用一个 enzyme 叫做 dehydrogenase， OK， 上面有 E1、E2、E3 那个鬼东西有没有？那个很大一坨的那个，OK。So guess what? Your pyruvate dehydrogenase is inside of the mitochondria. We just saw that. Okay. So wait a minute. You are going to make fatty acids in the cytosol. Okay. But your acetyl-CoA is in the mitochondria. Right. So how do you get your acetyl-CoA into the cytosol to make your fatty acids? Right. You're going to have some sort of transport. Now, do you guys remember what acetyl-CoA looks like? And you're going to say, acetyl-CoA 不就 like 两个碳，然后这边一个 S-CoA 有没有？哎，对，但是你要知道 S-CoA 这一坨东西啊，那个 molecular weight 有没有？大概800。大概我忘记多少，800多还是700多？它是一个很大的分子 ，OK？ 那这个很大的 molecule， 你要叫它 go through membrane。Maybe not a good idea. Okay, maybe not a good idea. Okay, so it's not going to do that. So turns out what it's going to do is your acetyl-CoA. Okay, your acetyl-CoA. This is your acetyl-CoA from pyruvate. Your acetyl-CoA obviously has two choices inside of the mitochondria. Okay, 在 mitochondria 里面 acetyl-CoA 有两个 choice 哦，来咯，两个 choice 哦。一个 TCA cycle， 对不对 ？acetyl-CoA 变 citrate。第二个 acetyl-CoA becomes ketone bodies, and we talked about what ketone bodies are. Okay? Now, if it becomes citrate, it can then go either direction. It can either go through TCA cycle and generate NADH and ATP, or what it can do is it can come out. Okay? Citrate now is a small molecule, okay, relatively small, 6 carbons, not too bad. So it's a small molecule, and it has a transporter that can transport citrate out. So once you get citrate out, then this is, the white space out here is cytosol, okay? And in the cytosol, citrate can be split into acetyl-CoA and oxaloacetate, okay? Using an enzyme called citrate lyase, okay? Citrate lyase. It's gonna split itself into acetyl-CoA plus oxaloacetate, okay? And then acetyl-CoA can then be used to make fatty acids, okay? Now, oxaloacetate 要怎么回去？How does oxaloacetate go back to the mitochondria? 直接回去，会变 pyruvate， OK， 可以变成 pyruvate 回去，或者是可以变成 malate 回去 ，OK， 所以 pyruvate 要oxaloacetate， 因为它本身是一个 beta keto acid， 所以它并没有那么稳定嘛 ，right？ So oxaloacetate usually will become something very rapidly, okay? Becomes pyruvate or becomes malate, and then it can go back to TCA cycle, okay, in the mitochondria. Okay, this thing has a name, okay, called citrate shuttle. Okay, 没有很 creative 啦，就是它是等于用 citrate 出来嘛，所以就叫 citrate shuttle. Okay, so here's a detailed look of the citrate shuttle. Alright, so here, this on the left is your mitochondria. Outside is your cytosol. Okay? And what you have is inside the mitochondria, you have pyruvate dehydrogenase. It becomes acetyl-CoA. Okay? And acetyl-CoA can use citrate synthase. Citrate can then come out using a thing called citrate transporter right here. Okay, it's just a citrate transporter, a transmembrane protein that can facilitate the transport of citrate. And then citrate, once it's outside, okay, it's going to use this thing called the citrate lyase. Okay, it does not use citrate synthase. Okay, it's going to use citrate lyase. Okay, this is a minor detail, but citrate synthase. Do you guys remember what's the delta G for citrate synthase? TCA cycle 的第一步，它的 delta G 是很 favorable 还是没有很 favorable？ favorable， 对不对？没错，是 favorable。OK， 所以 citrate synthase，OK， 就是说拿 OAA 加 acetyl-CoA 变成 citrate 是一个很 favorable 的反应。OK， 那也就是说，当你如果要用 citrate synthase 把 citrate 切回去 acetyl-CoA 跟 OAA 的时候，这个东西就会变成非常的 unfavorable， 合理对不对？那这个 enzyme 为什么可以做到 ？OK， 这个 enzyme 为什么会做到？主要是因为它又放了一个 ATP 在里面。OK，I don't know if you guys saw this, but here you have an ATP input. Typically, a lot of citrate lyase, okay, that we see around, okay, actually what it does is it doesn't generate acetyl-CoA. It generates acetate and oxalic acetate. Okay, citrate lyase in like bacteria and in some fungus and yeast. Okay, usually they generate acetate, but in this case, what you can see is that we put in one extra energy. Extra energy will drive citrate to form acetyl-CoA and oxalic acetate, okay, which otherwise cannot be done, okay, because the delta G is like plus 30 or something. So we're good. So once you go to acetyl-CoA, and then acetyl-CoA can be used to make fatty acid biosynthesis, okay? And your oxalic acetate, like some of you guessed it, you have to can either become malate or it can become pyruvate, okay? And then you can use this malate alpha Kg transporter or pyruvate transporter to then transport malate into the mitochondria or pyruvate into the mitochondria. Here's the part that's kind of interesting, okay? What do I mean by it's kind of interesting is that if we are looking at fatty acid synthesis and fatty acid degradation, okay? So in degradation, you always cut out 2 carbons at a time, okay, at the very last step. Now, fatty acid biosynthesis, okay, is going to add 2 carbon at a time, okay? So, as we previously mentioned, the 3-keto-acyl-CoA thiolase, you should know what the keto-acyl-CoA thiolase is, which step it does in fatty acid degradation. 可以吧？最后一步嘛，好不好？就是 thiolase 嘛，就是那个，就是 beta keto 变成 acetyl-CoA 跟另外一个 acyl-CoA 嘛。Okay, all right. So your thiolase is very thermodynamically favorable, okay, in this direction. Okay, this is fatty acid degradation. Very favorable, KEQ of 10 to the 5, okay? 10 to the 5th power, it's very great. You know, this is one of the reasons why beta oxidation happens very nicely, okay? Now, you can then imagine if I want to do the direct reversal of this reaction, then what you have is a equilibrium constant of 10 to the minus 5. Okay, now 10 to the minus 5 means like you have to have 10,000 of this and 1 of that, okay, in order for this to happen. It's very difficult to do that. Okay, it's very difficult to do this unless you have a high amount of acetyl-CoA that can push the equilibrium, like in the synthesis of ketone bodies. Okay, right. Ketone bodies 就是用这个反应哦，就是把它拿2个 acetyl-CoA 然后推到 acetyl-CoA。OK， 所以这个反应不是不能发生，只是说它的 thermodynamics 呢不是很 favorable。OK， 那我们在做 fatty acids 的时候，可能我的 acetyl-CoA 的浓度不见得那么高，那我还是要可以做 fatty acids 啊，不然的话我的 membrane 就没办法做。Okay. Through evolution made it such that this reaction can become much more favorable by, instead of using acetyl-CoA, we're going to use this thing called malonyl-CoA. Malonyl-CoA plus acetyl-CoA give you pretty much the same thing. Okay. 现在这个 CoA 先 ignore 啦，我先只是给你看这个 chemistry. So you have this initially 3-carbon molecule plus a 2-carbon molecule. In the end, you get a 4-carbon molecule plus CO2. Obviously, this reaction is going to be very favorable. Why? Because even though the KEQ is still less than 1, okay, but CO2 leaves the system. Okay, CO2 is a gas, so it's going to readily leave the system, causing this guy to always leave, and then you will always push the equilibrium towards the right. 听懂意思哈？就是用 equilibrium 的话 ，CO2 会离开，所以你这边 reaction 就会 one way, one direction. So usually this reaction we say it's irreversible. Okay, pretty much it's irreversible reaction. Okay, and I can actually show you this reaction is irreversible because I did this experiment. Okay, it's you cannot do this guy plus CoA go back. Now, Malonyl CoA turns out, okay, the question is where do you get Malonyl CoA? Turns out Malonyl CoA is nothing more but acetyl-CoA plus bicarbonate, or basically CO2, same thing, and then using the power of ATP. Okay, so this guy, this acetyl-CoA guy, okay, plus bicarbonate, use ATP, you generate Malonyl CoA. And then Malonyl CoA plus another acetyl-CoA becomes the beta-keto 4-carbon CoA. Throw it away, CO2. And then CO2 can come in again to do this process. So the net, okay, the net input is just ATP. 看得懂意思吗？对，你看上面就是Malonyl CoA 加 acetyl CoA 嘛，但是 Malonyl CoA 其实就是 acetyl CoA 加上 ATP 跟一个 CO2 做出来的。那上面 Malonyl CoA 加 acetyl CoA 加出来，结果你还会吐一个 CO2， 那这个 CO2 就跟这个 CO2 是一样的 ，OK， 就一样的东西。也就是说，你在整个反应里面呢 ，OK， 跟上面不一样的是，你这边等于多放了一个 ATP， 就是这个 reaction 加一个 ATP， 就变成 fatty acid biosynthesis 的第一步。Does that make sense? Make sense? Okay, we're good. No confusion. Right? Now, the magical enzyme that does this is called the acetyl-CoA carboxylase, and obviously, you know, it takes in a carboxylate, and the substrate is acetyl-CoA, so it's acetyl-CoA carboxylase. Okay, duh. Right? Acetyl-CoA carboxylase is one of the most important enzymes, okay, because it is typically considered as the committed step for fatty acid biosynthesis. Actually, this is not quite true. I will tell you why. Right? Here it says acetyl-CoA carboxylase is the committed step for fatty acid biosynthesis. I wrote this, okay, I didn't copy it from a book. And then I say it's not quite true, okay, just like, dude, you're crazy. What do I mean? Okay, Malonyl CoA, you will see in subsequent lectures, Malonyl CoA can be used to make secondary metabolites. Okay, it does not mean Malonyl CoA has to be used for fatty acids biosynthesis. It can be used for other stuff. However, secondary metabolites, we don't need a lot of it. Okay, 你们有没有听过 secondary metabolites 这个名词 ？Metabolites 是代谢物嘛，对不对 ？Secondary 就是二级代谢物嘛。那二级代谢物主要什么东西是二级代谢物？例如 antibiotics，OK， 就是你不需要这个东西来存活，但是有这个东西可能可以给你一些 survival 的能力。比如说，你今天是一个霉菌 ，OK， 然后我会可能做一些抗生素，那我就可以把其他细菌干掉 ，right？ 我可以把附近的细菌杀死。那这可以让我提高我存活的能力，但是我这个 fungus 本身如果不生产抗生素，会不会死？不会 ，OK， 所以这就是 secondary metabolites，OK 的一种 ，right？ 那通常我们要做 secondary metabolites 的量都不会太大，合理啊？你没事做那么多是要干嘛 ？OK， 你可能只要一点点，你就可以把附近的细菌杀死，你不需要说做一堆啊。做一堆也用不到，所以通常那些反应或者那些pathways 的碳通量都不会太大，因为它们的 carbon flux 都不会 too large. So as a result, that's why I say acetyl-CoA carboxylase is typically the committed step for fatty acid biosynthesis, right? Because the flux used to make other metabolites usually is not as high as fatty acids, but just so that you know, okay? And also, this is believed to be the rate-limiting step of fatty acid biosynthesis, okay? Now, what I want to show you now is ACC, or ACCase, as sometimes we would call it. ACCase is a multifunctional enzyme with 3 enzymatic activities, okay? So it's an enzyme complex that has 3, I shouldn't say enzymatic activities, but it has 3 enzymes, okay? 3 functional enzymes on it, right? Now, the bacterial ACC, okay, for example, E. coli ACC, is a complex of 3 proteins, and turns out one of them is actually a complex of 2 proteins, right? So really, it's encoded by 4 separate genes. That's E. coli, right? So E. coli ACCase is coded by genes ACCA, B, C, and D. Okay, so ACCA, ACCB, ACCC, ACCD. Okay, that's E. coli. Now, in higher organisms such as animal cells or eukaryotic cells in general, like yeast, all this ACCase is one large single multifunctional polypeptide. Okay, so through evolution, they made a fusion of itself. Okay, so it's one gene called ACCase. Okay, one gene for eukaryotes, but for prokaryotes, it's usually separated into many genes. Okay, these 3 proteins. Okay, these 3 proteins are 1, biotin carrier protein. Okay, you're going to see biotin again. Why? This thing is a carboxylase, and biotin is a CO2 carrier. Okay, so it's the prosthetic group that we use for carrying CO2. Okay, biotin. And then we also have this thing called the biotin carboxylase. Okay, it's going to put the CO2 onto the biotin. And then we'll have this thing called the trans carboxylase. We'll see what that is in just a minute. Okay. 好，来这边跟各位讲一个小东西 ，ACCase 啊，你看哦。bacteria 的 ACCase 跟 eukaryotic cell 的 ACCase 明显，我这边就跟你讲不一样 ，OK？ 因为 bacteria 的呢是不同的 protein 组合在一起的 complex，eukaryotic 的 ACCase 呢是一条 protein，OK？ 那 bacteria 跟 eukaryotes 有这个不同的时候，会让这个 gene 成为一个很好的什么 target？design 抗生素的 target。OK， 为什么？因为它这个 enzyme 对生物来说是必要的 ，right？ 因为如果你这个 life 要长，这个 organism 要长，你就必须要有 fatty acids， 因为要 membrane 嘛，要 membrane。那没有 membrane 的话，它就没有办法 grow。那 membrane 是 fatty acids，fatty acids 从哪里来的 ？OK， 从 molonyl-CoA 来的。那 molonyl-CoA 是从 acetyl-CoA 来的。这个 enzyme 就是专门把 acetyl-CoA 变成 molonyl-CoA，right？那我们如果又知道说 bacteria 的 ACCase 长得跟比如说我们的 ACCase 不一样，那你是不是就可以去设计一个 I don't know enzyme inhibitor 去专门 target bacteria 的 ACC， 然后不要去碰我们的 ACC，right？ 那这个就会是一个 antibiotics，right？ 所以现在很多大家在研发药物啊，很多人还是花很大的资源在研发 antibiotics，OK？ 因为抗生素，这几年有一些被研发出来的抗生素可能才出来，比如说5年，然后就会有抗药性了，所以抗生素就一直不断的在跟 evolution 在比赛，就是说我们能不能开发出一些新的 antibiotics？ 那这个 enzyme 是一个很好的 target， 因为你一旦把 bacteria 的 ACCase inhibit 掉，那你的 bacteria 就会死掉。那因为 bacteria ACC 跟我们的 ACC 形状一定不同，因为光是 the fact that 它们是有 subunit 的，然后我们没有，所以你可以想象，我们可以设计一个药物专门去弄 bacteria， 然后不会伤到我们。Okay? So this is what happens in ACC. It's the 3 proteins and the 3 domains, and I'll show you what they are. Okay. There are 3 proteins. What are the 3 proteins? The first protein is this biotin carrier protein. Okay. So it's just a protein that has a biotin on it. Okay. Of course, you need to have another enzyme to put biotin on this guy. 合理吧？就是 biotin 不会跟它 spontaneously 的 react 哦。就是没这回事，对，就是你这个 biotin carrier protein 这边是一个 lysine， 然后 biotin 是在这边 Okay, so you have another enzyme that does this. Right? I'm not going to talk about that. But this thing is usually what people call biotin carrier protein, or BCCP or something like that. Right, so that's first protein. The second protein is the biotin carboxylation. What it's going to do, okay, is it's going to put bicarbonate, or essentially CO2, whatever, and use energy from ATP, throw the bicarbonate onto biotin. Okay? You throw it onto biotin. So that's why it's called the biotin carboxylase. Right? It's the thing that's getting carboxylated is biotin. Okay? And then the next enzyme is a trans-carboxylase. Okay? Trans-carboxylate meaning moving, like trans is transfer 意思嘛, moving my carboxyl group to a different molecule. So I'm going to move it from, here is a biotin CO2. I'm going to throw it, make it become acetyl-CoA CO2. Okay. Acetyl-CoA CO2 is called malonyl-CoA. Okay? So I'm going to throw the CO2 onto acetyl-CoA. Okay? That's why it's called trans-carboxylase. Okay? I transfer the carboxyl group. Now, after you do that, your biotin is back to the normal biotin on the protein, and then you got malonyl-CoA. The mechanism works like this. Okay? It's fairly simple. All right? Now, in order to put bicarbonate or CO2 onto anything, okay, most of the carboxylases that we talk about all require ATP, right? Except one. Two. 我们讲过哪两个 carboxylase 不用吃 ATP？ 给你第一个是 PEP carboxylase， 它是 PEP 到 OAA 直接吃一个那个 carboxylate， 你直接吃一个 bicarbonate， 然后不用 ATP。那原因是因为 PEP 的能量本来就很大，对不对？所以 PEP 就已经等于是 pyruvate 加 ATP 了嘛，对不对 ？PEP， 所以它 PEP 的能量本身就很大，所以它不用吃 ATP。另外一个什么 carboxylase 我没有讲过不用 ATP 的，就是不是那个上面1/3讲的，是最近可能2个礼拜前。啊？有没有要猜一下 ？carboxylase 哦 ，carboxylase 是拿来干嘛的？插 CO2 在东西身上嘛，对不对？那我们前阵子才讲了一个什么跟 CO2 有关的？什么什么叉叉叉叉叉 carboxylase / 叉叉，有没有 ？Rubisco， 没错，是 Rubisco， 它叫做 ribulose 1,5-bisphosphate carboxylase / oxygenase， 有没有 ？Rubisco， 它也是一个 carboxylase， 它不用 ATP。Okay? But most carboxylases require ATP. Okay? And this is actually the mechanism of, similar to most carboxylases. The way it works is actually very simple. You have hydrogen carbonate or just bicarbonate. Okay, so this guy's bicarbonate. Okay. Bicarbonate, it has a negative charge. Okay. And if you have a negative charge, that means it's a good nucleophile. This guy is a nucleophile. Okay, nucleophile because it has a minus charge. And then it's going to attack a electrophilic center, and typically electrophilic centers are like carbonyl center, or in the case of ATP, your phosphor center, because this guy has a lot of oxygens surrounding it. So the electrons from this guy is drawn away. So this P is usually a good electrophile. So nucleophile is going to bang attack there. Okay? And then you generate this thing called the carbonyl phosphate. Okay? So it's just, this is the CO2 part. Okay? This is the bicarbonate part, and that's just a phosphate. Okay? And you spit out ATP. Right? So you activate, and you use ATP to activate your bicarbonate. Okay? And you're like, 那为什么这个不叫什么 like bicarbonate kinase 之类的？ 没有嘛，这只是一半而已。如果有一个 enzyme 专门把你 bicarbonate 变成 carbonyl phosphate 的话，那它可能就会叫这个东西了。Okay? Alright. Now, once you form this carbonyl phosphate, then this particular carbonyl center is also a good electrophile. So what you'll do is you will use your biotin right here. This is biotin. And biotin has these two nitrogens, and nitrogens have a lone pair of electrons. So lone pairs, typically this thing is also kind of a good nucleophile. And either nitrogen is okay, whatever, but probably usually this one, because here you have more sterics on the left. And these lone pair electrons will attack your carbonyl center, attack, okay? And then what you will get is, so 你的这个 nucleophilic, you make a nucleophilic attack onto the carbonyl center, right? And then your electrons go up, come down, phosphate leaves. So the mechanism is always like that. You have a nucleophilic substitution. Okay? You substitute a nucleophilic something, and then you kick out another group. That phosphate is a good leaving group, right? Because phosphate, once it leaves, it has resonance. It becomes very stable, right? So it leaves, right? And your biotonated enzyme becomes carboxy biotinyl enzyme. Okay? We're good. We're good. Now, the next thing is you're going to have acetyl-CoA come in. Okay? Now here, this guy is acetyl-CoA carbanion. Carbanion 是哪来的 ？Before 这个东西可以有 resonance， 它是怎么来的？它是一个什么 carbon？ 这个 carbon ion， 这个 anion 在什么 carbon 身上 ？alpha carbon 身上，对不对？那 alpha carbon 它的那个 hydrogen 的 pKa 够低，可以让一个 general base 把它 remove 掉，就这么简单。OK， 所以这个 mechanism 我们也看了很多次了，就是 carbonyl 旁边的那个 alpha carbon 身上的 hydrogen 可以很容易的被一些 base 去 remove 掉，然后你可以形成这个 alpha carbon 的 minus charge。那这个东西可以有 resonance， 有没有？所以它这个够稳定。OK， 那 even though 这个很稳定，它这边还是一个 minus charge， 它 electron 还是很多，所以它还是可以去做一个 nucleophile 这样。OK。And then 你会有个 general base 在这边，它把 proton 拔掉，然后 electron 就会变成这样嘛。那这个东西会有 resonance， 可以变这样。Okay. Now, because this is a good resonance structure, so this thing is stable. Okay. But a lot of electrons. Okay. So what it's going to do is it's going to do a nucleophilic attack on this carbonyl center. Okay. And electrons come up. When it comes down, pop out this guy. Okay. So same thing. Just like what happens in here. What happens in the second step. Pretty much you have a nucleophilic substitution. You substitute the nucleophilic group. Okay? You do that, then you made a formal bond between your initially negative charge right here with the bicarbonate or COO minus, okay, carboxylate. And then your biotin is regenerated. Okay? We're good? 有没有很开心？没有，对不对？我也知道。Okay, now, like I said, your fatty acid biosynthesis enzymes are different between bacteria and eukaryotes. Now, what I want to show you here is that in bacteria, each enzyme of the fatty acid biosynthesis pathway has its own enzyme. Okay, so we moved beyond ACCase. Okay, so the next part, what we're going to talk about is the pathway to make fatty acids. Okay, now, you're going to have 1, 2, 3, 4, 5, 6, 7 different enzymes, okay, for bacteria. And then for eukaryotes, you also have these 7 enzymes. However, in eukaryotes, these enzymes are merged into a single mega enzyme. Mega enzyme 听起来好厉害。Mega enzyme complex called fatty acid synthase. Okay, it's just called fatty acid synthase, right? So it looks like this guy, okay? And it's a huge enzyme, like huge, literally huge, and it has different domains, okay? So functional part of the enzyme that can catalyze different reactions, okay? And if you look at the enzyme, it's one single polypeptide from the N terminus to the C terminus, okay? That has pretty much all of the above domains, okay? Now you guys should understand how malonyl-CoA is synthesized using ACCAs, okay? We're good. Now we're going to talk about the core portion of fatty acid biosynthesis, okay? Meaning, what are these guys, okay? What are these guys, okay? Now, so fatty acid biosynthesis, the first step, okay? The first step is what you're going to do is you're going to transfer CoA onto this thing called ACP, okay? You're going to transfer whatever that's on the CoA to ACP, right? And you're going to have two things. You're going to have a primer, okay? So fatty acid synthesis primer, and a addition unit. Addition unit is always malonyl-CoA. So every time when you add 2 carbons onto the chain, you're always using malonyl-CoA to add 2 carbon. However, before you have anything, you have to have somebody to add to, okay? So in that case, you can use acetyl-CoA, okay? So what happens is this. You're going to use the malonyl or acetyl-CoA ACP transacetylase or acetyltransferase or azotransferase. You can call it a couple of things. You can either call it transacetylase or azotransferase. Pretty much the same thing. You know, they're transferring the acyl group, right? And which is why we call it AT domain, azotransferase, okay? AT domain. And the AT domain, it's going to load or it's going to transfer whatever that's on the CoA to ACP, okay? So let's first take a look at the acetyl part. You're going to have acetyl-CoA coming in to the enzyme, and this enzyme is acetyl-CoA ACP transacetylase or transacetylase. You will transfer the acetyl group onto ACP, okay? And then you have acetyl ACP, right? Now, what you notice is that both CoA and ACP, they have the same thiol. So the reaction is actually fairly simple. It's just a transesterification, okay? 你们在有机应该有听过那个 transesterification 的一个 reaction， 就是一个 ester， 然后你换那个 R group， okay？ 用一个 alcohol 去换。那这边呢就是 thiol esterification, I guess。那你就是拿一个不同的 thiol， 不同的 SH 的那个 group 去换 ，OK？ 那这边它是这样嘛 ，OK？ ACP 是一个 thiol， 然后它可以去换 ，right？ And once you get to acetyl ACP, next thing is going to happen is you're going to use this beta-keto ACP synthase. Okay, so this is really the what we usually call like the first reaction, okay, of like chain elongation. You know, the first reaction for chain elongation. Okay, so what this guy will do is it's going to then transfer the acetyl portion onto the Ks. By the way, Ks stands for keto synthase. Ks, keto synthase. And then your acetyl portion will then be attached to the Ks also at the thiol. Okay. Now, on the protein, you have a thiol. Which amino acid do you think it's from? 然后就点点点点点点，对 cysteine, okay, cysteine. Right, so this is active site. You have a cysteine. Okay. All right, so that portion is done. Now malonyl-CoA has to do the same thing. You're going to use this thing called the malonyl-CoA ACP transacetylase, and you're going to transfer the malonyl portion from CoA onto ACP, and then you generate malonyl ACP. Okay, so for fatty acid synthesis, remember I said it's different from degradation. We don't use CoA. Okay, we're going to use ACP. But before you get ACP, they're all CoAs. Okay, so you convert CoA to ACP. Okay? Okay, huh? All right. So at this stage, you set up your acetyl portion. Okay, so this is like the primer for fatty acid synthesis. Okay, and it's already on the enzyme. And then here, this guy is the building block. Okay, or the extension unit. Okay, so this is the thing that's going to be used to add to people. Okay, huh? All right, we're good? Let's compare acyl carriers, okay? Let's compare ACP and coenzyme A, okay? ACP and CoA both contains this phosphopentathione moiety, okay? Now, this pentathione thing, okay, is vitamin B5. Okay, so that's one of the reasons why we eat vitamin B5. I think it's B5. I think. 好像啊，我印象没记错的话。Also, this thing is used to make coenzyme A, right? We talked about that when we talked about CoA, right? Now, what is different and what is similar, okay? The similar part is both of them have this arm, this phosphopentathione moiety part. Okay, so this guy and this guy. Between ACP and coenzyme A, same thing. Okay, so really their chemistry is almost identical. The only part that's different is in CoA, here you have a AMP looking thing, right? So you have a adenosine, phosphorylated. Whereas in ACP, it's a protein. This guy is a protein right here. Serine, okay, it's a protein that has a serine attached to your cofactor. All right? Now, your CoA and E. coli's CoA and trees CoA, same thing. 大家的 CoA 都是一模一样，化学结构完全一模一样，大家都是 coenzyme A。因为 coenzyme A 它是一个这个小分子，不算小，但是是一个小分子 ，OK， 它是一个 molecule。那这个 molecule 呢，它就是这样子的结构 ，coenzyme A。But ACP is a protein. A protein is coded by a gene. Okay, so your ACP and E. coli's ACP and plants ACP may or may not be the same. Make sense? 懂意思哈 ？Right， 就是 ACP 它是一个 protein，OK， 所以 protein 可能会有不同的 shape depending on 它的 sequence. But of course, shape 不会差太多啦 right? And usually it's a quite small protein. In case of E. coli, it's like 77 residues. It's not big, but it's not like tiny. Okay, 77 residues. It's still roughly 300 base pairs in terms of DNA sequence. Okay, like 200 something. Okay, make sense? All right, so we're good with this. Okay, so the difference between ACP and coenzyme A. This is the fatty acid biosynthesis overview. Right, so on the last slide, we know we got this acetyl KS. So you loaded the acetyl group onto the KS, onto the ketosynthase, and then you're gonna take malonyl CoA coming in. Okay, and the reaction is actually quite simple. Thiols, okay, so these things are good leaving groups, kind of. Okay, so they can leave. And what happens really is your malonyl ACP here is carbon 1, 2, 3, and on the third carbon, this guy is a acid. Okay, it's a carboxylate. Okay, now if you look at this thing, it's kind of like it's very similar to a beta keto acid kind of molecule, right? Because if you say this is the acid, this guy would be alpha, this guy would be beta, right? So it's similar to alpha beta. It's like a beta keto acid. Okay, now, so what we're gonna do, okay, what happens is that the negative charge on the carboxylate will collapse down, okay, and you form a double bond like this. Okay, so this is the same thing as CO2, okay? And the electrons here are going to go to the carbon. And here you generate CH2 minus. Now, carbon ion, why is this thing stable? 为什么可以做这个 resonance? Okay, again, same thing as before, resonance. Because this is your alpha carbon. Alpha carbon is okay to have a charge, minus charge. Okay, because you can share the electrons out. Now, this thing now is a very good nucleophile because it has a minus charge. All right, and this minus charge is going to attack the carbonyl center right here. Okay, because this guy is a electrophile, and this guy is a nucleophile. Okay, and when you take the electrons here and you attack it to carbonyl carbon, electrons pop up, pop down, and then this electron here pop over there. Okay, and your Ks, of course, you can grab a proton in the solution, and then you generate your Ks, and Ks is attached to a thiol. Okay, and you also liberate CO2 here, and then your molonial ACP becomes the beta keto acyl ACP. Right, so it's the acetyl part on the Ks gets added to molonial ACP. OK， 所以不是当两个1加1就是你管谁是谁加谁嘛，对不对？但是它其实是这一段加在它身上，你变出来以后呢，这个就是比较后面，这边是头，这边是尾，看得懂吗？懂我意思哈？哎，因为我们待会要讲那个加4个碳的时候，你就要想一下说是谁在加谁了。OK， now once you get this beta keto acyl ACP, beta keto, next thing is beta hydroxyl. So you use NADPH, simply reduce it, and you're going to use an enzyme called keto acyl ACP reductase. Okay, why is it called reductase? Because the substrate is keto acyl ACP. Okay, 所以它不会叫 keto acyl ACP dehydrogenase， 因为这个东西没有东西可以 dehydrogenate。那你是拿它，然后去 reduce 这个，所以就是它是一个 reductase， 专门来 reduce 它。另外，如果这个 enzyme 一模一样的 enzyme， 你也可以叫它另外一个名字，叫做 hydroxy acyl ACP dehydrogenase， 它就会往那个反方向。OK， 所以你要搞清楚名字，名字有时候会告诉你它的 direction。这样 reductase blah blah reductase 就是一个 enzyme， 它拿去 reduce blah blah。OK， 可以哈。Alright, so you have beta keto acyl ACP reductase. Now usually we call them KR, 就是 keto reductase. 我们上面有一个 keto synthase KS domain， 这个是 KR domain. Okay, so KR. 如果这个 make 你的 life easier， 你可以这样子 remember 啊，好不好？就是如果我问你一个问题，然后你就写 KR， 我不会算你错啊。但是麻烦你还是知道一下它全名啊，不然就是一天到晚 KS KR 的。Right? Now, once you generate the beta hydroxy, what happens next? Okay, beta hydroxy, the next thing you're going to do is a dehydration. Okay, so this thing is going to call a 3-hydroxyacyl ACP. I'm talking about this guy. 3-hydroxyacyl ACP dehydratase. So usually we call it the DH, dehydratase. Okay, so DH. And what that guy does is you remove the OH and you generate the alkene. So the next thing is, this thing is called the trans-enoyl ACP, and then we're going to reduce alkene into alkane. Okay, so we're gonna use enoyl ACP reductase. In short, ER. Okay, so we're gonna use the ER protein, and then that will turn this alkene into alkane. Okay, so basically saturated bond. Right? So once you have this guy, butyryl ACP, it's saturated. So then what you can do is you can do the next round of carbon addition using the KS again. Okay, and then it's KS, KR, DH, ER, and then that's it. Keep on going and going and going. Okay, so now what you notice is that always this chemistry is always something. Okay, you go from keto to hydroxyl, hydroxyl to alkene, alkene, you can go to an alkane looking thing. Okay? Now every time you see how going from no hydrogen to hydrogen, so you reduce this carbon, one hydrogen, same thing, nothing changed. Here's two hydrogen, so you do another reduction. Now, so from this direction, this is the fatty acid synthesis. And really what this is a reduction. Okay, so again, synthesis is reduction, okay, because we are making a bigger molecule. And making a bigger molecule, basically you have to reduce it. Okay, make more CH bonds. You're reducing it. Degradation, this is degradation. Okay, and essentially it's oxidation. Okay, so I want you to get this straight. Okay, now TCA cycle, we saw this identical thing. Okay, this identical chemistry: succinate, fumarate, malolate, ethyl acetate. Okay, so in TCA cycle, in the second half of TCA cycle, you are oxidizing, you're degrading, right? So you're going in the degradation route, which is the oxidation direction. If you're going in synthesis direction, you're going to reduce the compound. Okay, now obviously you should know, okay, from here to here, you need to have electrons, 2 electrons. Okay, from here to here, 2 electrons. Okay, and 2 electrons usually are mediated by what carrier? NADH, NADPH, or FADH2. Okay, so after you generate your butyryl ACP, you can continue to do the next round and the next round and the next round and the next round. Now, typically until the carbon chain length reaches 16 carbons. Okay, when the chain length reaches around 16, sometimes you can stop at 14, but most of the time we stop at 16. When you stop at 16, when you get to 16, you have an enzyme called thioesterase. Okay, right, which we call TE, thioesterase. This guy is an ester. Okay, this guy is a thiol ester. Okay, an enzyme that splits this, like cuts this in half, is called a thioesterase. Okay, thioesterase. So it's an enzyme that cuts thioester. Right, so thioesterase. Okay, thioesterase. Okay, so when the carbon gets long enough, it's going to cut it, terminate it, cut out. Once you cut out the ACP, then fatty acid is released. So you're not going to go through another round of fatty acid synthesis. Okay, very good. Maybe good. All right. So this is, whatever is on the slide. It's essentially this, right? So in degradation, okay, in beta oxidation, the first step when you go from saturated to unsaturated, you kick out FADH2, okay? And hydration, you don't change, you don't use any electrons, right? You don't change the oxidation state. And then when you go from OH to ketone, you spit out NADH. Okay? And once you get the alpha beta, this is beta keto, then you can do a thiolytic cleavage. This guy is called thiolase. 你不要把 thiolase 跟 thioesterase confuse 哦，不要闹哦。thiolase 是 thiolase 哦 ，thioesterase 是 thioesterase 哦。OK， 可以哈。那 thiolase 呢是把 acetyl-CoA 切出来，跟这边是 CN-2 的这个 CoA。OK， 所以这是左边是 degradation。那右边呢是 biosynthesis， 你会发现到说它们中间的 intermediates pretty much the same thing. Okay, the intermediates in the middle, same thing. Now, instead of using acetyl-CoA as the direct addition unit, you're going to use malonyl-CoA, okay, and you do a condensation reaction such that the malonyl-CoA part, okay, the malonyl-CoA, this portion, is added to here. Okay, so it's added to the front. Okay, it's not added to the back, it's added to the front. Okay, and then generate this. Okay, so whatever this guy is, it's going to pop up at the front. 懂我意思吗？可以哈。所以 malonyl 的那个 part 会出现在前面，然后原本这些东西呢会出现在后面。Okay, and then you can do NADPH reduction. And remember, stereochemistry is different, dehydration, and then another reduction. And then you get your straight chain acyl ACP. Okay? You're good? All right. Now, in eukaryotes, like I said, we'll have one single enzyme called the fatty acid synthase. And if you look at this figure, what you see is some of the stuff that we use. For example, KS domain is right here, KR domain is right here, DH domain is right here, ER domain is right here. And you also have this AT, MAT domains, okay, for the first transferring CoA to ACP, right? The MAT, this thing. Right? So you can see that in fatty acid synthase, all of the functional enzyme activities are all there in one single polypeptide, okay? And oh yeah, by the way, this is ACP. That's pretty crazy, huh? So ACP is right there, right? So ACP is also on the enzyme. Now, this is a very exciting figure, okay? So I'm going to show you what it really kind of looks like, right? So you have like ACP in the protein, and what the ACP has to do is it's going to swing around your carbon to different domains for reaction. 这个东西跟什么很像？这一招跟什么东西很像？ Pyruvate dehydrogenase， 对，它跟 Pyruvate dehydrogenase 其实很像，就是你有一只手，然后你就把你手上抓那个东西来抓来抓去，丢来丢去这样，然后丢到不同 domain 上去做 reaction。Okay， now first thing first， okay， 这是 very first step。你有 ACP， 然后 ATKS 这样子 ，OK， 所以这是空的 enzyme，empty enzyme， okay， just enzyme right there. But of course, ACP has been modified by vitamin B5 and that kind of stuff. Right? First thing first, you're going to have the primer for fatty acid synthesis. The primer being acetyl-CoA right here, and your acetyl-CoA it's going to use the AT domain. AT domain will react acetyl-CoA and then transfer that onto KS. First of all, it's going to transfer that to like ACP to KS. So it's going to use AT domain, transfer the acetyl part from acetyl-CoA onto KS. You have this malonyl-CoA guy coming in, and now you're going to use the MT domain, malonyl transferase, okay? MT domain, and then this MT domain is going to transfer the malonyl part onto ACP. And then your KS, acetyl on the KS, is going to react with the malonyl on the ACP. They react through the decarboxylative Claisen condensation. Whatever, 名词不重要，就 condense, right? Condensation. Right? And then you generate this thing where you have the beta keto acyl ACP. Okay? It is the acetyl part that adds to the malonyl part. Okay, so you freed up your KS. 这边就变 free 了。Now you have this beta butyryl ACP, which is beta keto acyl ACP. And then your ACP, this arm thing, is going to swing this guy onto the KR domain, and then you're going to reduce it. So what you see is initially it's a keto group, now it's a hydroxyl group. Okay, so it's been reduced. And then it's going to swing from KR group to the, by the way, 这边应该 DH, DH domain. Okay, DH domain, and then it gets dehydrated, becomes this double bond right here. And then you get swing from DH domain to the KER domain, sorry, not K, ER domain, the enoyl-CoA reductase. And then you reduced it to single bond. And then once this guy is a saturated acyl ACP, okay, you will use the AT domain again to pass this entire thing onto the KS. Okay, so now it's on the KS domain, and then next thing, this thing and this thing, pretty much the same thing. Okay, right? Pretty much the same thing, and the next reaction is going to be the same thing. Okay, so here 2 carbons, here 4 carbons, but it's going to do the same thing. Okay, so it does not matter how many carbons you have here. What matters is really the alpha beta position right here. Okay, that's the only part that's important. So once you do that, you're going to use the empty domain again, and then you're going to go to here. You again load malonyl-CoA onto the ACP. Okay, you load malonyl-CoA onto ACP, and then it's going to do KS, KR, DH, ER. Okay, it's going to repeat. This is fatty acid synthase. So all happens at one enzyme. And then you're going to ask me like, 那你的那个 TE domain 在哪里? I'm just not showing you on this picture. Okay, so when the carbon gets to C16, 16 carbons, that means that it's large enough that your TE domain is going to be very active towards this chain length, and it's going to cut it off, and then you have a fatty acid come out. So this is, that's it for fatty acid biosynthesis. Now then the question is, okay, 大家说你要吃什么鱼油啊？为什么鱼油里面，或者是你要吃什么橄榄油啊？橄榄油里面有那个非饱和性脂肪嘛 ，right? Unsaturated fatty acids. Right, so the next thing we're going to talk about is unsaturated fatty acids. OK, 然后你心里在 OS 是，我有真的那么 care 吗 ？OK, 可能没有，但是不管啦，你要 care. OK, 好 ，unsaturated fatty acid biosynthesis. All right. Now, unsaturated lipids are very important for various cellular functions. Okay, so in us, okay, you'll see in just a minute that unsaturated fatty acids participates in a lot of immuno responses and to make certain hormones or certain biological sensory, not sensory, messengers, okay, for transferring different important messages across the body. Okay, now. Also, for a lot of different organisms, remember that unsaturated fatty acids will increase the fluidity of the cellular membrane. Do you guys remember why? 为什么 fatty acid 就是这种 unsaturated fatty acid 可以 increase 我们叫做 fluidity， 就是你的那个 membrane 的那种流动性，有点像柔软度啦 for membranes. All right, remember unsaturated fatty acids because the natural unsaturated fatty acids always cis, okay, cis alkene in the cis conformation. If it's in the cis, you can see how the structure is going to look like. It's like you have a little kink, a little, KK 的就这样子会转弯嘛 okay? So when you have a bunch of these cis fatty acids together in a membrane, they're supposed to pack. But because of the spatial organization of this thing, they don't pack very well. ，right？ 他们在 pack 的时候，你就可以想象说，一堆如果是长碳链的一堆一堆，他们就可以接的非常非常好。这就为什么那个饱和性脂肪，像那个我不知道你们有没有中午去吃那麦当劳，或者有时候去吃麦当劳的时候，你有看到他们那个员工就那个要在油锅里面要补那个油，有没有？他补的不是油，是一坨东西，对啊，但是那一坨就很像就奶油啦，就是他拿一个很大的一个塑胶桶，然后就开始哇，就拿那个你们做实验那个瓦冰块的那种东西有没有？就哇一桶那个奶油就直接倒到它的油锅里，那一坨奶油那坨油啊，那个 butter， 那其实那一坨油就是因为它几乎全部都是饱和性脂肪。那在 room temperature 下呢，它们的叠的非常非常好，所以它会呈现固体，会呈现 solid 的。But 这个 alkenes， 如果你这边 alkenes 很多的时候，都是 cis fats，right？cis lipid， 那 cis lipid 它就会弯来弯去，会导致它们在空间上的 annealing， 就是结合的时候，会不是那么好卡。也就是这就为什么它们的 melting temperature 会往下降。那 melting temperature 一往下降，在室温下，它们就是油体，就是 liquid state， 就不是 solid state。那当然，你不要说那种傻说什么新闻说，那个这个什么橄榄油对身体好没有？然后你回家想要炸薯条来吃，你就用橄榄油去炸薯条，不 OK？ 因为那个这个油会高温，它高温嘛，你可以想象高温，然后你这边有 alkene， 它可能会去做一些反应，反而不好。所以炸东西确实是要用饱和性脂肪，这就为什么他们说那个炸的不要吃太多，因为你会吸收一堆这种 saturated fats。那 saturated fats usually 它没有什么 special function other than 能量储存 ，OK， 所以就只是 generate fat 而已。那这种非饱和性的 unsaturated fats usually have other functions，OK，so for fishes，OK， 在像那种深海鱼啊，为什么它们的那种 omega 3会特别多？因为它们要活在深海里面，那温度通常不会是什么很舒服的25度 C 嘛，可能 usually 会很冷。那在那么冷的环境下，它如果身上的那个 membranes 或者是它的脂肪，如果是 saturated fats 的话，那你可以想象它就是一坨东西在那边，不太合理，对不对？所以它们需要很多的 unsaturated fats。可以让他们的 membrane 以及他们的脂肪可以 still move around， for 那种深海鱼这样 ，right？ So unsaturated fatty acids are important for a lot of times animals or organisms that can grow at cold temperatures, right? Then the question is, how are these unsaturated alkenes made? Because we talked about fatty acid biosynthesis, and every time we add acetyl-CoA, and you always get saturated, okay? You always get saturated. You can't just use this guy and start the next round, okay? You can, okay? You can, okay? 有 proteins 可以那样做，但是不是拿来做 fatty acids, okay? Okay? So once you get saturated fatty acids, how do you generate the unsaturation? Okay, the enzyme they're going to use is called this thing fatty acyl-CoA desaturase. Right? So desaturase is to make unsaturation in your alkane, okay, in your fatty acids. Now, just look at the part on the left, and you're like, okay, I'm going to put an alkene there. That should be simple. I can go from alkane to alkene very easily, right? Now, the problem is actually, if you look at this oleic acid or even this palmitoleic acid, where's the alkene? It's in the middle, okay? In the middle, you don't have your carbonyl next to it. You cannot deprotonate. You cannot possibly deprotonate these guys, okay? So then you're thinking, wait a minute, you have these alkanes. How do you do reactions on them, okay? So you're not going to use a normal fatty acid enzyme. Okay, you're going to use a special type of enzyme, and this type of enzyme will actually use molecular oxygen as the oxidant. Okay, because molecular oxygen is quite oxidized. It has a lot of oxidative energy. Okay, so we're going to oxidize this thing using O2. Okay, that's the first thing. You're going to use oxygen to oxidize your alkane. All right. And this we will talk more about how to use oxygen later in discussion of amino acids. But just understand that some enzymes can use oxygen to oxidize things. Now, what you should notice, okay, what you should immediately notice is that going from alkane to alkene, how many electrons did you lose? 2 electrons, right? O2 to water. How many electrons is that? 那你这时候就要回想到我们讲 photosynthesis 的 light reaction, okay? Right, it's 4 electrons, 对不对？我们说 light reaction photosynthesis 不是我们可以把水变成 O2 吗？然后我们不是就是把2个 water 变成1个 O2， 然后不是会有4个 electron 飞出来，然后4个 proton 飞出来，所以是4个 electron。那 this molecule you lose 2 electrons. However, for molecular oxygen, you're gaining 4 electrons. Okay, so the electrons don't balance. Therefore, actually what happens is that you have to use NADPH to supply the other 2 electrons necessary to have a balanced reaction between O2 to 2H2O. 你从这个 saturated single bond 到 double bond， 这边是2个 electron 的落差，这个 OK， 对不对？但是从 O2 到2个 H2O， 这边是4个电子的落差。OK， 所以这边吐了2个电子，吐给谁？吐给 O2， 因为 O2 到 H2O 是 gain electron，right？ 因为我们说光合作用的时候是 H2O 到 O2 吐 electron， 所以如果是 O2 变 H2O， 那就是吃 electron。OK， 但是从 O2 到2个 H2O， 我要吃4个 electron。但是这个分子只能让我拿到2个 electron， 那我还少2个 electron， 那这个 electron 你就需要用 NADPH 来给你，不然的话你这边就没有一个 balance 的反应。Turns out this enzyme you have to use NADPH as well. Now it's actually very interesting because then you're considering like 那这个 enzyme 它到底是在做 oxidation 还是在做 reduction， 对不对？它又做oxidation， 但是它又吃一个 NADPH。OK， 那当然我们 usually 呢是在看它 main 它主要的那个 substrate 的这个 direction，but just so you know， 它是一个这样子的反应。OK， 所以 always 要记得哦 ，O2 变2个 H2O 呢，中间是4个电子在不见。Okay， 但是我们一般的 oxidation 跟 reduction 是2个电子，2个 electron。Right? So you use the power of molecular oxygen to then generate alkenes in the middle of your fatty acid. Then if we look at the pathways of polyunsaturated acids, okay, this is mono, right? 1 alkene, so this is mono unsaturated. Okay. What about poly? How do you generate polyunsaturated? Okay. So pretty much the same thing. Okay, you're going to do the same thing. You have a delta 9 desaturase. You will have a delta 12 desaturase. You can have a delta 15 desaturase. So you have different desaturases that act on different carbon positions of the fatty acids. Now, mammals, mammalian cells, or like at least us, we only have delta 5 and delta 6 desaturase in addition to the delta 9. Okay, so if we look at plants and animal on the very top, for both plants and animal, we can do delta 9. Okay, however, only plants can do delta 12 and delta 15, and then mammals can do delta 6 and delta 5. Okay, 那 delta 几，那就是从 carbonyl 开始数第几个碳上面会有 alkene， 那就是 delta 几这样。Okay， 那 what you should notice is that animals cannot generate 靠尾巴的 alkenes， 就问题就来啦 ，omega 3有没有？常常听到大家说要吃 omega 3，right？ omega 是怎么算的？是从后面算，对不对？是从这个尾巴开始1、2。假如说这是 fatty acid， OK， 1、2、3、4、5、6、7、8、9、10、11、12、13、14、15、16。OK， 假如说这是一个 C16， 是有听到什么东西吗？哦 ，OK， omega 3，OK， omega 3， this is omega 1、2、3，OK， 在3的地方 这边呢会有 alkene。OK， 那就是1、2、3、4、5、6、7、8、9、10、11、12、13。哎，我有少碳是不是？whatever 啦。By the way， 不是 trans。Like that. Okay. So， 等我一下哦。So basically towards the end. Okay. So if you look at this chart right here, if you have 18, delta 9 and delta 12, right? So the bigger the number is, meaning the closer it is to the end of the molecule. Okay. Now only plants can do 12 and 15, so we can't do that. So omega-3 we can't make. Okay. So we have to get it from other sources. Now you're like, wait a minute, then how does fish do it? Okay. Fish eat a lot of like seaweeds and that kind of stuff. Okay. So in their diets they have a lot of omega-3. Okay. So and then we eat those fishes, we get omega-3, so we're happy. Right? Now, what's important is that once you get these unsaturated fatty acids, like for example from plants, okay, we need them, right? So we actually absolutely need these things. We need these polyunsaturated fatty acids so that we can synthesize some important intermediates. Okay, so what you see is you have a bunch of different unsaturation, elongation, whatever. You don't need to memorize that, but just so that you know, we need to have these polyunsaturated acids. So on next Tuesday, I will quickly finish what these polyunsaturated fatty acids are used for.